Hi, how are you?
Good, how are you?
Good. Centers, 10:45. Singles or groups?
Oh, single.
You by yourself?
Single.
Single.
All right. Who's my moderator for today? Do you want to get them started and get their PowerPoint pulled up for the first one? Oh, wrong room. That's why that doesn't sound right.
I think it's already open.
Monica, are you by yourself? You can press that.
I'm presenting by myself. I don't know why.
Okay. All right, so you're up first. We have a minute or two, but you're up first.
Okay.
Have you used the clicker before?
I am not.
Did they tell you to use the clicker? Svetlana, is this room live? Svetlana, this room's live and ready to go? There's nothing we need to click on. We can just start presenting.
The Zoom meeting is already on.
Okay.
Zoom is on, but live, Juliet turning from her side. I think she's turning at 10:45, so if it's not time yet.
It's 10:45, so
Okay,
I just
okay,
wanted
let
to
me step and just double check if,
wait for her. Wait a minute or wait for her. I'm pinch-hitting right now, so I apologize if I don't sound as intelligent as a faculty member who actually does do this. If you want to switch to a history presentation real quick, I'd appreciate it very much. Annika, is this your 290 project?
Yes, it is.
everything is live, all the rooms, so you
All
can
right.
start.
All right, hello everyone. My name is Anthony Miller, and this is my research project on isolation and characterization of novel viruses that kill E. coli. And I was mentored by Dr. Nick. So my first question I'm going to start with are what are bacteriophage? So this is the novel viruses that we were studying. They are viruses that infect bacteria and kill them either through the lytic or the lysogenic cycle. So they require a bacterial host to reproduce, and that reproducing is what kills the E. coli. And they are completely harmless to humans, as their bacteria only kills those bacterial cells, or as their DNA only kills those bacterial cells. So they can be specific to a bacterial strain, or they can kill multiple species. The phage that we were looking for were very specific to 4 strains of E. coli, and they were non-pathogenic human E. coli strains. And then for every grain of sand on Earth, there are over 1 trillion phage. So they are extremely prolific. They're everywhere around us in every piece of soil, and they can be extracted fairly easily, as we did in this research project. And they can be visualized to the naked eye using plates of bacteria with plaques of phage. So the plaques are the little clear circles you can see over there. This is on a K12 strain of E. coli. And you can see a few different morphologies. So those are like the different sizes and shapes of plaques. So there's a few small ones, some big ones, and this is one of our original plates of E. coli that we work with. So here's some background more on bacteriophage. So there's the structure of it. It has an isosahedral head, and that's what holds the DNA. And then the tail and the tail fibers. The tail fibers is what recognizes receptors on top of bacteria, and that's how they know which bacteria they can reproduce with. So this is an emerging field. Bacteriophages were only discovered about 100 years ago, and research into them only started about 90 years ago. So it can be used against non-pathogenic and human pathogenic strains. With the phage that we discovered and characterized, we can go on to further testing with human pathogenic strains of bacteria, but usually it starts on the ones that are less harmful to humans. And they're extremely diverse in morphology, which could possibly lead to diversity in usage. So this is our purification process. It's fairly simple and can be broken down into 5 broad steps. So we started with collecting a sample from the ISU soil farm out by the teaching farms. And we took that soil, purified it, and separated the liquid that can contain phage and ran it through a 0.22 micron filter and plated all of that liquid onto 4 different strains of E. coli. With those plates, we picked each distinguishable morphology and we isolated them to grow them separately. When we grew them separately, we did it with a spot test. So that confirms that there's actually phage in the pick that we got, and that it was specifically the phage that we were looking for, hopefully just one morphology. And then once we could confirm that we have one phage there, we flooded the plates with some phage buffer and amplified it, ran it through the 0.22 micron filter once again, and then we had our vial of pure phage. So here's our process just in a little more detail with some pictures. Here's a soil farm up top with heaps and heaps of steaming soil. It's out there at all times. And we put it through the 2.22 micron filter, which you can see in the middle right there, and plated it on those original 4 plates. So each of those letters represents one strain of E. coli bacteria, W, C, K12, and B. As you can see, we got some really good plaques out of K12. We can see some distinct morphologies there. C, we can't really see morphologies, but there was a lot of phage that killed that. W, we have kind of a foggy one plaque that we tried to pick. And then B, we ran into a problem with the bacteria itself. But due to the time constraints on this research, we decided to just move forward with the plaques that we could distinguish the best. So K12C, and we tried on W a little bit. So once we would pick those, we would put them in these little centrifuge tubes right there with some phage buffer. So it was tiny, tiny amounts of liquid holding like millions of phage that we could work with. So we would pick them, dilute them, and put them on plates like this. So here's another C plate where you can see the morphologies more. We had 3 on that one. And we ran into some problems with C down the line as well, which I'll go into a little later. But really it was just time constraints. The more time you have with a project like this, especially involving phage, the more discoveries you can go through. But we were on a very short time schedule through this semester. And then all the way over there is our final 4 purifications of our phage. So K12 worked the best for us throughout this entire experiment. So we really just moved forward with those. And those are the 4 vials that we sent to the electron microscope to get pictures of. So here's the setbacks I was referring to. As you can see up top, we tried to pick a few phage and plate them. And the C strain of E. coli, we don't know if there was a contaminant or if it wasn't strong enough to build a whole wand, but we couldn't get anything from that. And we, again, just moved forward with the K12. The W, we tried to pick that really foggy plaque many, many times. So that every time we did, we had a contaminant in it, as you can see in those like discolored circles right there. So we were going to plate the phage that we got at the end of the experiment on all of the strains anyway, which is why we could just continue moving forward. It wasn't really about finding a phage that was specific to a strain. We wanted to find phage that could kill multiple strains. So again, as I said, more times, more phage discoveries. So with our results, we strive to answer 3 questions. Are there phage, which was determined by the purification process? What do they look like, which is determined by the electron microscope? And what can they kill, which is a plaque assay against all of the host strains. So here's our first phage and the pictures that we got with the electron microscope. So as you can see, there was a long tail. It was fairly plentiful in the purification and fairly strong, as it could kill 3 out of the 4 strains of E. coli, K12, C, and W. You can kind of see up here the different rings in the tail. There's some emerging research about the rings go maybe pointing towards more bacteria or more DNA in the head of it, but there's nothing kind of concrete yet. But these are some really good pictures that we got from that. And then phage B. So we never really got a picture of a full phage in the electron microscope. We only got these like shell resembling blobs, which points towards an extremely low concentration, especially since it could still kill 3 out of the 4 strains, K12, C, and W. So some repeat imaging on this would be helpful to see what kind of morphology you have. Phage C, again, we didn't really get any good pictures. This is a like really zoomed out one, but we couldn't find anything which points towards either low concentration or a contaminant, especially since it could kill the K12 strain, which we had already confirmed through the purification process, and it was very weak against the C strain. So something was going on with this phage. Again, repeat imaging would be helpful, but since it could only kind of half kill something other than its like host strain, we wouldn't really move forward with it. And then phage D, which is turned into phage D/E, turns out we had 2 different morphologies of phage in this concentration. So you can see there's one that has the long tail like phage A, and then we got a really good picture of some of the short tail ones that we had in this. So you can see the kind of horizontal end to those tails. That's the tail fiber that will recognize the receptors on top of the bacteria. And you can kind of see the like rings on it as well. They were both really plentiful in the concentration, and they were able to kill K12 and C strain together. There's some ways to isolate these. We could dilute it a little, look for little changes in the morphology on the plaques, do some genetic imaging, a lot of different things we could do to purify it. But again, time constraints, and they worked well together. Most times that phage is used to kill or to as a therapeutic, it's in a cocktail anyway. They're never really used by themselves. So this isn't necessarily a bad thing. And we got some really good pictures. So you can see like the deflated heads of a few of these. So that means that the phage has already inserted its DNA into the bacteria. So it's really great that we got a picture of that. So here's the plaque assay I was referring to before. As you can see, A and B killed all different strains. C was really weak against some, and we had a contaminant in D, but you can still kind of see the halo around it on K12 and C. And then, sorry for crossing, but as you can see here, it's a little like foggy around the edges of that and more crisp over by W. So these are some really, really great spots. In the foggy part, that's the phage performing the lysogenic cycle, which is when it integrates its DNA. And then the lytic cycle is the more clear thing. So it was really great that we could see that that type of phage performs both. So what next? Further imaging and testing on phage A and B because it could kill all 3 of those strains. And the testing would involve human pathogenic strains at the vet med lab. And the novel therapeutics is just the phage cocktails I was referring to before. These things are really used as an antibiotic alternative or a last resort, especially in cases where the patient has like things like MRSA, where antibiotics just don't work anymore. And sequencing genomes is really helpful with phage, so you can distinguish them from each other. And there's a process where you can take out the part of the genome that performs the lysogenic stage and turns them kind of into like assassin phage that work really, really well against bacteria. And then the industry needs a lot more funding and resources to properly rival antibiotic research. So the industry is fledgling, I would say, but I think it has a lot of promise. And thank you for your time. Any questions or anything?
Can you go back? Slide, go back one more. So, yeah, go there.
Yes.
So when we're looking at this and we're looking at the contaminants you keep referring to, is the contaminants in the process of collecting the phage, or is the contaminant something that you're picking up through the process of purification? Have y'all been able to identify that, or is it just something that is naturally occurring in?
So it's picked up during the purification process. We use like all plastic things that were run through like a sterilization, but since it wasn't glass, we couldn't like sterilize it every pass through that we opened it. So there's a lot of chances that just a different bacteria can get into it. Sometimes we get lucky and the phage kills that as well. In this case, we weren't as lucky. And usually you can see the kind of halo around it, but you can't really pick from that, which is why we couldn't go forward with the W's. It's unfortunate, and we didn't really go forward with the ones with contaminants because it's such a long process to try to separate that phage from it in the first place. And that kind of bacteria, the contaminant, also kills E. coli, or at least grows on top of it. So it's just a setback that it's unfortunate. We didn't really plan for it, but it happens in labs. There was like a lot of people working in the lab as well. The masks probably helped, but it's just with the plastic. Yeah. Is that answers your question?
What's the next step for your research?
So I asked Dr. Peters to be my capstone mentor. So I'm going to do my capstone project with him. I really like lab work. I'm an animal science major, so this isn't completely in line with it, but I'd love to do some like phage research with more animal-centered diseases instead of human pathogenic ones. And just go forward on phage research. I'd love to do this like multiple times a week. We only did it once, which is the time constraint that I was referring to, but as far into this as I can go, I think I would love to do that. Yeah.
All right. Any other questions? Thank you. Cool. Yeah. For purposes of presenting, I should have said this before you started. The presenter is allowed, if you're standing over here by the second slot, to remove your mask if you're more comfortable with the presentation. And to keep you into the video, if you're standing over here by the second window shade, you will be in the video as you're speaking. And I'm sorry, I turned my computer on late to get that on for you. So thank you, Annika. That was very informative. Let's see, who's up next? Is that William?
I'm William.
Okay, is William not with us?
That's
Oh, you're William.
Am I up next?
Yes. If you would please. Fetch a pipeline. Is that what I'm looking at?
Yes.
All right.
Am I in the shot?
We're not.
Oh,
I don't know why we're not getting you here.
I just saw the blue X on the floor and I thought,
Yeah, that's what I thought it's supposed to be, but it doesn't look like I have everybody else in the shot. Come a little further down. Yeah, that's a blue X, but it's not showing you in the,
which way, this way?
yes. There you are. There, you are in the shot now. We got you. You look beautiful.
Yes.
All right, so please introduce yourself and tell us so the people online know what you're doing.
Hello, my name is Bill Winnett. Today I'm going to be presenting Fetch, a pipeline to parse NCBI, the genomic data, and organize it in multiple forms. And it's a little tool I've been working on for the last year or so, since about approximately the beginning of the pandemic. I've been advised and assisted by Drs. Dennis Lavrov and Raj Muthi from EEOB. Yeah, let's get started. So some background for a case that you aren't necessarily a biologist by trade or anything. There's many different fields that use genetic sequences, protein sequences, all that kind of things, such as biology, genetics, bioinformatics, biotechnology. That's just a few, but there's many others. Depending on the type of, or the arrangement of those bases inside that sequence determines a completely different arrangement of life. And that great diversity of possible combinations leads to the great diversity that is available in life. My lab works with mitochondria. Typically, I orientated this whole project towards the mitochondria, but it's not necessarily limited to just the mitochondria. But it is a little bit more specific and niche towards genomic tools. Inside, or for every individual, there's the arrangement of like genes like that, and that all combines together to make one genome. It's the arrangement of all genes inside that organism. To put it in perspective, these things are remarkably similar, but yet different. For example, like us in the room, we have about 0.1% difference in genomic composition, but between us and a banana would be about 40 to 60%. That's a heavily debated figure, and if you were looking for research or somewhere to go into. But we can also use that information and determine what's the same, what's different, to infer like how it's evolutionary different, or how it's adjusted along that line. The other thing I'd like to mention before I get into my project too is NCBI. It's a gene bank, which is a government-funded database which houses millions of sequences. If you ever need a sequence or anything, just go to the gene bank. If you need an article for things, you can just go there too, and it's a lot of times it's free. And the key thing I want to hit home for this is every individual thing on the gene bank is cataloged, has its own record, has its own tags, has everything to identify it. So that's what fetch comes in from. You could go to the gene bank and manually get it yourself. I just don't like doing that. It seems like a pain to do. You have to hit a certain kind of button, you have to hit a certain kind of format. And if you have more than one, it's also pretty difficult. Like you have to do it multiple times, or if you're using different tools such as BLAST, you could just hit a button and like get it all. But sometimes you're not doing that. You just want to get a couple of them off, which is where fetch comes in. The other reason why fetch comes in too is that especially in bioinformatics, you get used to working with the command line or the terminal, and fetch can be ran just from that. You can simply type in the exception numbers you need and fetch, and then it'll pull it off all down for you. The other thing too is a lot of other projects or a lot of other tools that you might use require a different format. Like it has to be all in a certain format, or everything has to be conglomerated, and fetch does all that for you. Or most of it, depending on what else you need for it to do. So if you need for it to be a whole genome or a single translated gene, that's that covered. So there's a couple of different segments to fetch. First part is fetching, which is very aptly named, I suppose. It's to automatically retrieve things from the gene bank. So all you would do is type in fetch, then the exception number, type in what you would be searching on the gene bank, such as Drosophila as a tax ID, however you want to enter it. After that, you'll download it and store it into a couple different schemes. One is the gene bank file, which is what you would see as if you were just going onto the website to look at it, which is essentially just like a web page, just text format. The whole genome, a full FASTA, which means that like every single gene is added to one single document and saved as per species. And the individual, which means that every gene is pulled apart and you get a copy of your own individual gene in case you just need that one gene for whatever reason. The other thing it does too is it translates all of that depending on what the translation table is that was posted. A lot of this assumes that people are, whenever they submit a genome or submit a sequence, that it's properly annotated. On the right, I have a picture of the folder scheme. It's kind of just puts a little bit more of a graphic to it. The storage just has several different styles of how it's stored. The output, we'll get into later. Indexes we'll also get into right now. Image didn't quite line up. But the indexes is just a simple record of what is there and what's there. What's not there, if it's not there, it wouldn't be on the list. Genes and species have their own separate lists. So you could open up the species index and see, I have downloaded Drosophila melanogaster, I've downloaded this sponge, I've downloaded this bacteriophage, what have you. Each record that you have would be marked with a semicolon by default. And the reason why is because if it's unmarked, then you're able to pull it out to a different location, which we'll get into in just a second. But it's just noteworthy to know that everything is by default with a semicolon in front and can be marked off for pulling. Oh, also, everything is refreshed automatically as soon as you were to fetch anything new. So pulling, you have all this information. What do you do with it? That's not really up for me to decide. That I'm just here to facilitate a scientist using their stuff. But if you're working on a project where you needed to know, or you needed to work to decipher something with ATP6, you could download a bunch of ATP6, unmark it, and then pull it all out to a file, then work with it after that. You'll have a whole collection of simple ATP6s. You can see how it changed. So another way to say it is pulling is just essentially whatever you have unmarked, pulls out every individual copy of that and consolidates it into one single copy. Every ATP6 gets consolidated, every COX-1, every whatever you have. Same with species, but there's also species individual files, so it's kind of vector. Pipeline terminus. This is just another way to say end. The point of all these pipelines are to be compatible with other pipelines. It's almost like a Lincoln log setup where you have one part attaches the next and so on. It's currently compatible with a sequence alignment tool, and that's it so far. But after that, there's stuff in the works to be added, 4 different other tools, which I have not used yet. But the point of it is not that I'm comfortable with it, but rather the scientist is that's using it is compatible with it. The other thing too is that it's really open-ended, such that if anything new comes out, that like say next week, some kind of great tool is released, it's all in the same format for that to be added. Other than that, just simple customization is all of the next step. That's fetch. Any questions?
So are you working towards this to be like publicly or commercially available?
It currently is.
Cool.
It's you have to download it through GitHub, but yeah, you just got to go to it, clone it, then it should be available as is.
So I'm going to ask a question that's a little, I'm not sure if I missed it or I didn't quite tie it in together. How does this fit? It's in your title, it says multiple forms based on BioPython. So is it the computer programming that you use? Is that what to do this fetching, or
Yeah, the whole,
is that how the BioPython ties into what we're looking at in the fetching up here?
it's built on BioPython.
Okay, so that's what, so it was the computer program the software that you were using to build it.
Yes.
Okay, that's what I was trying to understand. How, oh, go ahead.
Oh, it's good.
No?
Oh, okay. Because you saw that we can use on a command line and everything. So if I want to fetch a gene from a specific species, do I need to have specifically the accession number, or could I just type the name of the species and the name of the gene and that will fetch, or you need the accession number?
I feel like it'd be more efficient to use just the accession number, but you could also do the species and the gene. It works the same way as if you're typing onto NCBI. It's like it's literally the same exact.
Okay, so if I type on NCBI, the name of my species and the gene, and whichever results I get there, I will get with fetch.
Yes.
Okay.
The other thing to mention too is that NCBI recommends that you use like Boolean operators and like tax ID, the species, and all that kind of stuff. I would recommend that too. Just makes it cleaner.
Okay, thanks.
Other questions? Well, thank you very much. That was. All right, so Andrew, we're
Yes.
now going to transition to you.
Thank you.
If you feel the need, there are lights out wipers there, if you feel the need, if you need to clean off the, and we share.
All right, I gave you a contaminated.
No, you're good. Yes.
I'm gonna ask, can we slide you over here a little bit? That way, it makes sure the line of sight is there, over there, out of sight. In chair.
Thank you, I appreciate it.
You guys see me or am I in the screen?
Yeah, just give us a second, so, there's a lag on Zoom.
Oh, no
So
worries.
it takes a minute, so right now, you've got your beautiful assistant assisting us. All right.
Okay, good to go.
Mhm.
All right, so my name is Andrew Lavin, and I will be presenting on cadmium and its ecotoxic effects on demethylation in Chrysemys picta turtles. And without further ado, let's begin. So, brief overview, I'm going to go into the background of the supplementary info for our experiment. I'll state our hypothesis, I'm going to go over our results and methods, finish with some conclusions and implications, and then open the floor for closing statements. So, sex determination, it's what establishes the sexual fate of embryos. And if we look at this, there's two types from this reference. Okay, so there's GSD, which is genotypic sex determination, and that's like us with sex chromosomes. And then there's ESD, which is environmental sex determination, and that can be influenced by a variety of environmental factors. So what decides the sex ratio in natural populations, and that's important for ESD especially because that usually forms a 1-to-1 ratio for males and females. And in some taxa, sex determination can be influenced by temperature. And then the Shoemaker and Cruz, they do a really good job of illustrating this. So an example is like for males, let's say a lot of the male development genes are catalyzed by the SRY gene on the Y chromosome in the mammals. Whereas in a case of temperature sex determination, it would be catalyzed by a temperature that's inducing that species. And then so C. picta, our little turtle that we're working with, is a species that utilizes temperature sex determination. And just to kind of reiterate on PSD, which I refer to as temperature sex determination for most of this presentation, during development, males and females have different patterns of expression for sex-related genes. And then what we're going to be going into right now is that one way of affecting gene expression is the presence of DNA methylation, which is an epigenetic mechanism, means it alters from the outside. So what exactly is DNA methylation? So DNA methylation is when a methyl group replaces the 5' carbon on cytosine, and it's usually followed by guanine. So if you look to this figure to the right, we'll say like CPG, that's what it's commonly denoted as. And the figure kind of helps give a really good overview of the basics of DNA methylation. So if you look at that top figure, we're going to see that if it's unmethylated region of the promoter, our transcription factors are able to bind, and as a result, gene transcription occurs, which means gene expression occurs. And on the inverse, if there's methylated regions in the promoter region of that CPG, the transcription factors are unable to bind, which means that there's no transcription occurring and no gene expression. So therefore, DNA methylation is commonly seen as a repressor for gene transcription. And in past studies, we've seen that C. picta has shown a possible link between its TSD and DNA methylation. And the slide just kind of reiterates on that. So to this right is a heat map that is for C. picta hatchlings that were incubated at different temperatures. So we've seen denoted as male 1, male 2, female 1, and female 2. And we see in each of those lines on there on the C map, some of them are kind of hard to see. It's technically a gene that they were studying. And what we see is in the areas of low methylation, those genes that have low methylation activity, we see that those areas in the female are a lot of high methylation. And it goes inverse. In the areas where there's a lot of low methylation, which could mean that there's gene expression for those female genes, there's a lot of high methylation in the male. So yeah, we see this inverse DNA methylation pattern between the males and females. So temperature is just one factor that can affect sex determination. Another is the introduction of contaminants in the environment, and these are commonly known as ecotoxins. And the ecotoxin that we focused on was cadmium, which is a heavy metal ecotoxin. It's primarily common in agricultural fertilizers and just even commonplace fertilizers. And the problem with it is that C. picta can be exposed to cadmium in the natural nesting sites due to agricultural and irrigation runoff. And previous studies have shown a link between ecotoxins and possible C. picta gene expression, which if you look at that bottom reference right there, they ended up applying a topical solution of cadmium to C. picta embryos. And what they had found was they actually found traces of cadmium in the gonadal tissue, which is the sex tissue of the embryos. And they also saw a stress induced response, which meant there was genes that were being actively upregulated due to this presence of this ecotoxin. And then, yeah, so previous studies have also shown that cadmium could also affect DNA methylation. So our hypothesis, cadmium could influence the DNA methylation in the gonads of developing embryos in Chrysemys picta, potentially influencing the sexual development of these turtles. And how exactly do we go about doing this? So our experimental setup, we first treated C. picta embryos with cadmium of a topical solution, which was 1 microgram per gram of egg. And then we would place these at 26 degrees Celsius, which is the male producing temperature for C. picta, and 31 degrees Celsius, which is the female producing temperature for C. picta. Then we finally collected DNA and RNA from embryonic gonadal tissue through extraction. So first we performed a PCR assay, which is the polymerase chain reaction. And this was just to test the methylation status. And this was done after doing a digestion restrictive enzyme. And so the primers were designed for DMRT1 and aromatase, and I've bolded them because they're going to come up a lot in the results. So it can just help to kind of have this in their head. But DMRT1 is a male development gene, and it's known to be involved in testes production. And then aromatase is a female development gene, and it's known to be involved in ovarian production. So the results of the PCR assay, we used gel electrophoresis to visualize the results. And we saw that DMRT1 showed evidence of demethylation as well as aromatase. So there's a negative correlation between methylation and cadmium, meaning when there was cadmium was introduced, we saw less methylation. And so these gels are kind of hard to read because they're kind of loaded, but hopefully this legend kind of helps. So on this left side, we have our undigested samples, and on the right, we have our same digested samples. And on the top, there's males and females. So what we're looking at is on this left side, we're going to want to see 2 bands, and that means that there's methylation present. So we see that for stage 9 aromatase and stage 9 DMRT1. And then we're looking at our digested samples to see if there's a change in methylation. We want to see a fading of bands or only a singular band. So we see the fading of bands on stage 9 aromatase primarily, and stage 9 DMRT1 is really good because we see that we have a couple areas with just a single band. So this is showing that there was a change in methylation following the digestion and cadmium treatment. So the gel results continued. We see the same pattern for stage 15 DMRT1 and stage 22 DMRT1. And I just want to note this because when we're going into this, stage 15 is known as usually the beginning of sex determination, and stage 22 is known as the end of sex determination. Unfortunately, if you see our lovely gels on the top for aromatase, they didn't go out too well. This could be for a couple reasons. It could be primer design for the aromatase. It could have been a bad PCR or even just bad gel imaging. So unfortunately, at the time of this presentation, we don't have the same conclusive results that we do for DMRT1 or for aromatase as we do for DMRT1. But we're going to continue on. So we have a qPCR assay next, and this was performed to test the expression levels of DMRT1 and aromatase. So we're here where we're just testing like the qualitative change in methylation. Here we're actually not testing the expression levels of DMRT1 and aromatase following cadmium treatment. And we used 15 samples for each stage and temperature, and then a statistical analysis was performed using a junk t-test. So the results of the qPCR assay, we'll start with DMRT1. So if you look to this left figure, it's a control expression of DMRT1. And what we see here is what we'd expect to see for a male development gene. We see at near the end of sex determination, near stage 22, there's a higher expression level in male producing temperature at 26 degrees Celsius versus the female producing temperature. And now when we start looking at DMRT1 expression after cadmium treatment, we're seeing this almost inverse. We're now seeing that there's a higher level of expression for DMRT1 at stages 15 and 22 at the female producing temperature. And one thing to note, and I'll go into it once I finish up, I'm going to go over aromatase, but this not significant was for all 3, and I'm going to go over that in a minute or 2. But what we're basically seeing here is that there is a difference in the control expression. And kind of going over aromatase, we see the same thing for the control expression. Aromatase near the end of sex determination is higher at the female producing temperature, and we see this differential expression between the male producing temperature that drops considerably low. And now when we look at our aromatase expression after cadmium treatment, we see almost a similar thing that we saw with DMRT1. We see a higher level of expression at the male producing temperature at stage 15 and stage 22 versus the female producing temperature. And again, these are not significant. And that's actually really interesting to see because it's not significant. It's basically saying that we're not seeing a differential expression that we're seeing for these controls. So we see like we do see a definitive like differential control here, but when we start looking at ours, it's not. So this could be possibly showing that there's a disruption in the sex ratio. It's just because we don't have an influential like differential expression that's usually needed for temperature sex determination to form that 1-to-1 ratio. So the overall conclusion was there's a negative correlation between cadmium and DNA methylation. And this was primarily seen in the PCR. We saw that gene expression was affected by changes in DNA methylation patterns, which we saw with the qualitative control. And then we also observed demethylation at both genes. And that means we saw that consequential change in gene expression patterns through the qPCR, which we saw in these last slides, where we're not seeing that differential expression that we normally see in a natural population. And then finally, this could be showing evidence of possible genome-wide methylation rather than targeted methylation. Because we saw this at both sexes for both genes, it doesn't mean that a singular gene is being targeted by cadmium. It could just mean that this is overall a genome-wide effect of methylation patterns. And again, I kind of touched on it when I was going over the qPCR, but further implications, so the natural populations of the turtles could be affected by the cadmium exposure. If we're seeing this, no longer seeing a differential expression for these two like very important genes, because they're like are what forming the gonadal tissue, this could be disrupting their sex ratio. And we'll see piglet being a potential keystone species in its habitat. Because its sex ratio is being disrupted, its population can crash. And as a result of that, the other species that are in its habitat can also have their populations crash because of the absence of C. picta. And just like acknowledgements, I'd like to thank Dr. Nicole Valenzuela's lab. It's the lab I've worked in for the past 2.5 years. She allowed me to work on this project and has been guiding me through it. I also want to thank Bia. She came, she's my grad student. She basically got me through this entire thing. She helped me with the presentation throughout this whole experiment. And then the co-author, Nick, he did all the qPCR. He's also a grad student in the lab. And then he did all the qPCR data and testing. And thank you. Any questions?
So at what point in time does cadmium start to, so I'm trying to think through this. Is it embryo development while still in the turtle, or is it also something that's affected after the eggs have been delivered and buried?
So if I think I know what you're asking, so yeah, like how we set up the experiment was we touched the egg. So that's primarily how it could get into effect with them, because that's when the turtles would be the most vulnerable. It's like when they're in their eggs and as embryos and they're developing. And then I don't know if this helps answer, but I did kind of gloss over, but these were all like true with cadmium even from stage 9. So even though stage 9 isn't really the beginning of sex determination, the cadmium still was affecting them. So it happens throughout their entire embryonic phase.
Okay.
If that makes sense.
Other questions?
I mean, I know everything.
Thank you very much.
Yeah, thank you.
Okay, then that brings Jennifer up next. The problem is you can't see the slides on here. I don't know how to switch it over so you can actually see the PowerPoint slides. There's any way to turn the camera. Yeah. Give it just a second. Yeah. Does, will you have it, or were you waiting for Thelma to come in from,
Oh, she can.
yes, we haven't started yet, so we can invite her in.
You guys can come in. Me too. Are we still trying to figure out the slides?
No, we're not gonna, there's a lag on Zoom right now, so I was just making sure. I don't know how to get the slides to show up on Zoom, which is what they're asking for. But you're welcome to start at any time, Jennifer. Thank you.
My name is Jennifer Ortiz. I'm a senior majoring in biology here. And my project looks at the invaginated eye of disco clam and determining whether or not it uses opsin-based phototransduction to detect light. My faculty mentor on this project is Dr. G. Serb, and I've also been getting a lot of help from a postdoc in her lab, Dr. Kyle McElroy, both in the Department of Ecology, Evolution, and Organismal Biology here. Before I go and talk about my methods, the results, I'm going to break down some of the jargon that's in my title, just because it is a bit, especially if you're not familiar with it. So opsins, structure. Opsins are light-sensitive proteins that are found in most animal eyes. Phototransduction is the process of light being converted into an electrical signal, then sent throughout the nervous system, and then processed, and then the organism is then reacting to that light stimulus, however it seems fit. And then an invaginated eye is an eye structure that sits just underneath the surface of the tissue, and it doesn't directly interact with the environment. A really interesting thing about marine bivalves is that they have 5 different eye types. And so there's these nice little simplified versions of these eye types up top. There's a cap eye spot, pigmented cup, compound eye, mirror eye, and the invaginated eye, which is what our disco clam has. And another cool thing is that they're family specific. So as we see with the invaginated eye, which is denoted in this purple color, it's only found in family Limida, which I reference as limids later on. And this is where we can find our disco clam. So why is this work particularly important? Eyes are extremely complex organs, and they're also fairly diverse. They're used for navigation, interaction, and detection of environmental cues. Bivalves are particularly interesting because they have these 5 different eye types. And we can't assume that they're all using the same method to detect light if they detect light at all. And this is just further pushed by these images on the right. They just show A and B have the invaginated eye, so they're very hard to tell where they are. They're denoted by these arrows. And then the rest of the boxes show compound eye, if I remember correctly. So this study is being conducted to better understand how the disco clam specifically detects light, if it detects light at all. And what we take from here could be used in an aquacultural sense. We never want our organisms that we're kind of culturing up to be stressed in any way. So determining what frequencies of light causes them stress or has them in a less stressful environment would be best. We never want to be cruel. So the organisms that we use in this project are Argopectoderadians, which is a scallop that has been previously identified to use opsin-based phototransduction. So we use it as a reference, kind of a backboard to this project. We also use for our limids Tenoides ales, which is our disco clam, Tenoides mitis, as well as Tenoides scabber. So we added these 2 other limids to this list, mainly to have a bit of a more robust data set. And we're also curious to see if there was any similarities or differences within the options that we find here, if we find any at all. So because of COVID, my project has been mainly computational as well as preliminary. So going through, I'll be talking about kind of what we did at each step, as well as what we got from each step of our methods. So starting off with RNA sequence reads, transcriptome assembly, FLAS, motif search, and then our resulting tree. So I grouped RNA-seq and transcriptome together mainly because they go fairly hand in hand. The programs that are listed on the left are essentially what we use in order to build up our transcriptome. We were given raw DNA reads or raw RNA-seq reads from another institution who already did a similar project like this, but not focusing on opsins. They also gave us a transcriptome, but given the fact that it was a couple years ago that they made it, and we weren't 100% sure how they built their transcriptome, we wanted to take it into our own hands. And programs are always updating and changing. So we wanted to make sure that what we had was up to date. And so on the right, we just have a quick little section of a sequence just to kind of show you what we're working with. And we use these kind of sequences to turn it into a workable and searchable database, which is the transcriptome. From there, we use BLAST, which is essentially a searching program, a searching tool, using the argopectin irradians and their pre-identified opsin sequences. We took each one and searched them against the 3 limit transcriptomes in order to identify if they had opsins at all. And then we took those opsin results and blasted them back against the irradians just to make sure that what we were having was in fact opsins, or at least close enough to opsins so we could use. And then from there, we wanted to look at motifs, which are essentially short, repeated sequences that have been conserved within a particular region, and they have a structural and functional purpose. That's why they're being conserved. We use Eugene, which is a nice visualization and annotational tool. And so just a quick screenshot here of what that looks like with our species up top, along with the sequence following right after, and then a nice little snippet of where when you search up a motif, it flags it really nicely, and it also provides these little drop-down menus. And so after going through and looking at and identifying motifs, we have this resulting chart. So our species are on the left, so disco clam up top, and the argopectin irradians on the bottom. One thing to make note of is that color denotes structural or functional results despite the slight differences in sequencing. So in column RTC, which is an opsin type, we have FAK and ACK. So basically those two might not be exactly the same, but they have the same ending result. Because opsins are fairly unexplored in bivalves, we're not quite sure exactly what these guys are doing in terms of structure and function, but we know that they're conserved and they're present throughout multiple bivalves, specifically within the opsin gene. So we're hoping that further down the road, they can be kind of more explored so we can figure out what they're doing. Another important thing to make note of is that the RNA sequences that we were given weren't necessarily complete. So there are chunks that were missing in these sequences. So we couldn't identify all the motifs if there were more. And then finally, we have our resulting tree. It looks like a bit of a mess here. But one thing to make note of is that our sequences of interest, our limit sequences, are in color, and those that are not are in black. And we're just going to be focusing on this highlighted section because it really shows what's particularly interesting about this tree. So essentially, with the other opsin sequences from these bivalves, we noticed that they group by opsin type. So our first bracket shows OPS GQ2 kind of just clustering together despite species. And we see that with GQ3 and GQ4 as well. And then for our limits, we see that they group together by species and not necessarily opsin type. And there's a few things that could be determined from this. A few things to also make a note of. Our sequences aren't complete. So this could be very different once we have full and like well processed sequences. Another thing is that we identified these sequences and named them through what we identified them from. So argopectin radian opsin 1, and then the top result for that in lipids was named tenoides minus opsin 1. So it doesn't necessarily mean that they're the exact same gene type, but we just wanted to label them something and not have them just be kind of nameless opsins. But if this is true, and this is accurate, this shows us that these opsins are lineage specific and that they most likely diverge fairly early on in marine bivalve evolution. And that's really interesting. We didn't expect that. We would expect them to kind of file in with the others based on opsin type. And so future directions, because a lot of this was preliminary, we want to use different methods in tree building programs. We mainly used maximum likelihood. So getting in there and doing Mr. Bayes would be nice, just to see if there's any difference. Creating a more up-to-date transcriptome. We sent out our own sequences, or we sent out samples to get a full genome sequencing. So they should be coming in soon. So we can kind of just go back and like rebuild everything. And then determining what wavelengths limit opsins react to or absorb through photospectroscopy. So determining exactly what light frequencies they react to would be really cool. And so I'd like to acknowledge the EEB department, my faculty mentor again, Dr. Jean Serb, as well as the postdoc, Dr. Kyle McElroy, and then of course the ISU McNair program. And then I have questions. I also have my contact information up there if you want to reach out to me. But yeah.
So how do you guys go about collecting the data for like how the light is affecting the embryo?
Yeah, so this is a very like generic photo of me doing work, but essentially we have this cell culture just kind of constantly going, and then we take that opsin sequence and put it into that culture, and then we do light dark trials, so that insertion happens in the dark, and then from there we kind of put it in a cuvette, put it in like a giant spectrometer, and then we just run light frequencies through them.
Got it.
Do you expect to see extreme changes or different changes when you get the full sequencing based off the partial sequencing you have?
Originally, I thought we would, and then we kind of just file it together. But because of the fact that they are structurally different, and the fact that this preliminary work kind of shows that they're doing their own thing, I want to think that they don't like mix in with the other options. So I'd hope, my own personal hope is that they stay species specific.
Thoughts? Thank you very much. So thanks to all of our presenters, all four of you. You have done a wonderful job. We've learned quite a lot here today, a lot to be built off of from all the work that y'all have done. So congratulations. We will restart in this room after lunch around 12:30. So we will reconvene in this room around 12:30. So thank you all very much.
Thank you.
You weren't nervous, were
Yes.
you? You all seem very nervous. You all seem like you knew exactly what you were doing.
I wanted to like tunnel vision when I was presenting.
Well, the great thing is that most of us don't know, you probably have the most expertise in the room of what was going on, so, if it wasn't for the fact I've been sitting through these presentations for about 25 years, I wouldn't know half the stuff y'all are talking about, so great job.
Thank you. All right. Nice job.
Thank you too.
I was gonna have.
That's actually. No, I just started working, like, not like, I'm not like any bioinformatics stuff, but I just started working.
Well, when we saw that the door had the close sign on it.
Oh, no, because it was the coffee places there, and so they kept like, people were ordering things. and so a lot of noise, so
You couldn't see my face, but I was like.
I don't know if they want us to. I don't know.
So it looks like you lost the format.
Did you see Virginia? I think that's who it was who was in Danielle's session.
So you didn't sign up for the TRIO program, conference I gather?
Yeah, I did.
Oh, you did?
It's on my calendar.
The one tomorrow, the shot tomorrow.
Tomorrow and Friday.
Why are we getting a recon back?
I wondered that too, because I thought the way they had it set up, it seemed like it paid for our membership and what you call it,
Yeah.
the conference. Yeah, I don't know.
So I thought, did Ashley register or? Or you just-
And I saw for some reason Arnold's email was on there too.
I saw that too.
I didn't know what that was about.
Because I paid for mine myself. I mean, I don't know. And I've looked at the conference schedule and it's mostly pre-college.
Yeah, especially like the sessions and stuff like that.
So, anyway.
And I don't know how the camera set up person. This is they can talk about how because I know we asked about presentations. And I think, and they were like, oh, no.
Are you guys done in here?
Okay, yes, we are. So right now they're standing here and so. all
Okay, did you
the-
close your Zoom when you were done?
That's us. The other group just walked out.
Okay, let's just make sure we close the Zoom. Otherwise, we're gonna have just garbage on your stream.